but he will be joining us in a few minutes. So good morning, everybody, and thank you so much for being here at such an early hour. I will start with my short talk. And here is our modus operandi. After every presentation, there'll be an opportunity for you to ask some questions. And if we will have some more time, then we'll have a discussion afterwards. And here is our moderator joining us, Mr. Abramov. Please, can I have my slides on the screen? My name is Alexei Mazes. Thank you. And how do I change the slides? Do I have a clicker? Morning, ladies and gentlemen, and welcome to our conference called Healthy Moscow. As is tradition, we start very early in the morning, which means that we have a lot to discuss. It is such a pleasure to see our speakers that I believe have already been introduced. Oh, no, that is not the case. They haven't been. I'm with the University of Russian University of Friendship of Peoples, or the Rudan University, and we've established a HIV and AIDS center together with Moscow Center for HIV and AIDS prevention. Today with us we have Alexei Mazes, Chief Consulting Expert for HIV Diagnosis and Management. We also welcome Chinese investigator of Chinese Center for Disease Control and Prevention, Imin Xiao. We also welcome Mr. Margolis, Leonid Margolis, Senior Investigator for the section on intercellular interactions of National Institute for Health of the U.S. He's also with the Department of Bioengineering and Bioinformatics of Lomonosov State University, Jürgen Rockstraw, Professor of Medicine and Head of the HIV Outpatient Clinic at the University of Bonn, and Yelena Tsiganova, from the International HIV and AIDS Center at the Rudan University. Indeed, HIV is a challenge of the 21st century. The international medical community pays a lot of attention to this epidemic. For our university, this is indeed a special focus of our attention. It is a special focus of attention for our students, and we have students from 157 countries. Before, we used to focus more on some specific diseases or tropical diseases, but today in this interdependent world, we have these diseases that are considered socially important. Sometimes they are dubbed plague of the 21st century, and HIV is one of them. But indeed, that is not a death sentence for the patients. There are new modern ways of treating HIV positive people, and we will be discussing them today. And I wish you all a very substantive discussion, a very substantive exchange of opinions. I believe that our today's session will make its contribution to HIV management and prevention. Alexei, please, you have the floor. Thank you so much. Unfortunately, the clicker doesn't seem to work. It won't work, or does it? Let me try it once again.
О, хорошо.
Okay. To continue our moderator's opening remarks, some stats. Around 80 million people were infected with HIV since 1981. So you know what's quite unusual? We have our slides right above our heads. So you have to look up. Please get accustomed. What an unusual way of looking at the slides. But you see the statistics. Around half of these 80 millions have died. So I believe that on the slide you can see these numbers in square brackets, because up to this day we do not have a reliable measurement system. So these figures in square brackets, they tell you how variable the data is on HIV across the world. Unlike Russia, because Russia set very specific requirements for HIV statistics. We have a database of HIV positive people from day one. And you see this graph with columns showing you the latest data. 4 years ago, Russia has added to its database another set of data that represents those HIV positive people who are now receiving treatment from HIV centers. These figures are consistent with the statistics from Europe and the US. So these are patients who are being treated by HIV centers here in Russia. The figure is lower than our overall data, which is somewhat higher, but it reflects the real number of HIV positive people in our country. We started a little bit later than scheduled. That is why I'm somewhat in a hurry. What about Moscow? We've detected almost 130,000 people, HIV positive people, and the majority of them, they come from other cities and regions to Moscow to receive treatment. After that, after receiving treatment, they go home. But we have their data, we know them, and of course the hometowns of these people, they receive all of the data on the treatment of HIV positive people, which enables us to keep track of all of the HIV positive people in Russia. In the upper corner, of the slide, you see two trends. One of them is for Moscow, that is the blue line, the blue curve, and the red is for Russia. You see when the infection peaked, when a lot of drug users started to get infected. After that, the first HIV management program was launched, which enabled us to stabilize HIV prevalence. You can see that. And you see the decrease of newly detected HIV positive people. In Russia, we see the similar picture throughout the last year. But you see that some of the regions of Russia are responsible for a somewhat negative trend of HIV infection in Russia. The gray zone, the gray color that you see here, in the bigger graph, are people from other cities who came to Moscow for treatment, and you see that the number peaked in 2017. Now the figure is lower, but you can clearly see that non-Muscovites are bigger in terms of the number if we compare them to Muscovites who are HIV positive. That is a very important slide for us because it demonstrates how we work with young people. We see that we virtually curbed HIV transmission among young people. That is very important, crucially important. You see the figures and they really speak for themselves. You see the situation. The transmission type ratio changed. Back in 1999, we registered the maximum number of HIV positive people among Muscovites, and the majority of them back then were drug users. And we started back then to work with drug users, and that is why the share of HIV positive drug users in Moscow decreased compared to overall numbers across Russia, which again enabled us to curb the prevalence of HIV infection. Of course, in Russia and in Moscow, we see a large share of MSM people who are HIV positive. We see that in Europe, the majority of newly infected people are among MSM population, and I believe that Professor Rockstraw will be speaking about that in greater detail, about HIV and MSM population across Europe. We see the stats for the Baltic states, Latvia, Estonia and Lithuania, where the number of drug users among HIV-positive population is pretty big compared to other European countries. So you see here Moscow, Europe, Africa and US here in this table, and there are some serious differences in terms of cohorts of HIV positive people who need help and support that we offer to them. In Moscow, a large share of HIV positive people are drug users. In Moscow, just like in the majority of Russian regions, we have an interdisciplinary program to work with key populations. This program was endorsed by heads of Moscow government department heads. So that is how our management structure works. That is the Department for Healthcare of Moscow government who coordinates the efforts of various bodies. Social services work with us very actively. We work with the media, with various other committees. We work with teachers. We work with our Moscow Education Department, and indeed non-governmental organizations play a very important role when it comes to working with HIV-positive drug users. I know that I'm running out of time, so let me hurry up a little bit more. So that is how we work with HIV-positive people. If they receive treatment, they receive help from every department of the government of Moscow that work in this area. There are services that work every minute of their time. They provide beds for HIV-positive people. They are not within our remit, these services, but they are special services of various hospitals who work with HIV-positive people. The opportunities that we have today to help people living with HIV are absolutely enormous. Those of you who were there at the very beginning of our HIV fight efforts know how hard it was back then, and it is in front of our eyes that first ART regimens were established, were proposed, that helped HIV people to live. Later on, another medicines were designed to help people living with HIV live longer and more comfortably. Indeed, today we still encounter serious challenges when it comes to treating people living with HIV, and we'll be speaking about that today with Professor Rockstraw. And I invite all of you to this session because we'll be discussing how HIV is diagnosed, what it can be mistaken for when it comes to diagnosing HIV, and we'll be discussing how we can help people living with HIV. And indeed, we are very hopeful about the future, and today we will have a special session focused on new HIV vaccines, new drugs that will help us fight HIV. We'll be discussing prolongs accessing the markets. So what is the major challenge that we face today as a country, and what is the major challenge that every doctor faces today? What virus suppression a patient will have as soon as they have their therapy and they start to receive their therapy? If we speak about drug addicts, that according to data that you have on this slide, only in half of cases we can hope for a significant virus suppression. Speaking about Moscow, we have medical records of 23,000 HIV-positive people, but unfortunately around 5,000 of them do not visit our HIV center, and they still do not receive their therapy, and it is high time for them to do so. But unfortunately, they do not receive any treatment. And of course, they develop AIDS as the result, which is a very bad situation considering tools that we have at our disposal today. To try and see what are our options and what is our future when it comes to the UN AIDS program, we of course needed to find what we can compare ourselves to see a viral suppression in at least 90% of people living with HIV. And that is why we decided to compare Moscow with New York. Maybe Leonid Margolis will disagree with me, but I believe that in New York, HIV-positive people, they also have access to medications, they also have access to support and treatment. But the next slides shows us very clearly, and I would like to go to this next slide, please. Here it goes.
But this is not really true. It's really hard to achieve or implement that strategy even in New York. Yes, we treat more and more patients, but we cannot get the level of immunological suppression, especially with drug users. And some of those people get registered much earlier, and they are the MSM population. They register early, they are careful about their treatment regimens, but still, according to our data, we cannot say that we will not meet those goals of the UN AIDS program. We have some suggestions from that program, but once again, at this point, we don't see any real opportunities to achieve those goals. And this is my final slide that shows that overall, in Moscow, it's not all that bad. Basically, there's an interesting trend where in capitals you get higher infection rate rather than in the rest of the country, but it is not so. It's not true for Moscow, even though we have all features of a metropolis, of a major city, and we seem to have all the factors for high rates of infection, but it's not true. Thank you for your attention. I'm done with my presentation. All right, thank you, Mr. Mazus, and those were significant numbers and the achievements of Moscow, and they were made possible due to the support that we receive from the government of Moscow. Since we're talking about the multidisciplinary approach, I'd like to say that in the city of Moscow, many different agencies and government bodies try to tackle this issue from all different sides. And when they work together, we can achieve these results and get those great results. And that's why Moscow is a good example, both for Russia and other countries. And we have this international scientific or research and methodology center with the support of the Moscow Healthcare Department. We are a major center of scientific research. We study such matters as treatment and prophylaxis. Please pass your questions to our presenters in writing if you can. We will try to answer them. Let me give the floor to Mr. Yimin Shao, Chief Expert on AIDS, representing the Chinese Center for AIDS and STD Control and Prevention. Thank you, Yimin. You may have the floor.
First, I would like to thank the organizer, especially Alexei, to invite me to this very important conference. We know our two countries are neighbors, and also the G5 members. So we do fear the collaboration will have a lot of potential to grow and also to help our people to control this terrible disease. I would like to touch one area we have been doing for almost 30 years, is to try to use the network of CDCs of China. We have not only one CDC, we have 3,500 CDCs at the national, provincial, municipal, and county level. So we use this network to try to monitor the virus. Oh. This is not the slides I gave in this morning. These are wrong slides. Could you change to another file? I'll just give it to you. HIV intervention. Sorry. That's the vaccine talk. So we're being, at the early of HIV epidemic, I was in WHO, so in the 80s. At that time, yes. Could you move to the next slides? Yeah. We know HIV as an antivirus, it has reverse transcriptase, has a very high mutation risk. So the virus every day can produce 10 to the 10th virus with one amino acid difference. So we have a big power of genetic variation. Next slide. Yeah, I was in WHO, so as a young scientist at that time, almost 30 years ago, I was the only virologist. They asked me to do a virus research program for WHO, but everybody knows WHO is a bureaucratic agency. It don't have a single lab. So it took me several weeks to wrote a memo to suggest WHO to using the member countries' laboratory to build a network to monitor the HIV variation. And that's what the network did in the last 30 years, to identify the origin of HIV from African and how do they spread to different continents, affect the people. That's the major events has been identified at a different time of the year. I didn't stay very long. I stayed because the Chinese epidemic started. So I went back to China using the CDC network to monitor the HIV variation in our country. So in the 80s, we identified there is a clade B virus moving from the African to Thailand and then to our border region, Yunnan. You see from the left, the second peak, that is a border region epidemic. And then it became a second in the 1990s, a second peak. That is contaminated plasma donors. So that's the first origin of HIV epidemic in China. And then there's a virus from African to northern India and come to the border region again. It got quickly recombinant with this virus is clade C. And then it got recombinant with the pre-existing clade B and become a BC recombinant. The major one is CRF07. And then this virus travels from different regions of China and made the second largest epidemic. And the third one is a clade AE virus, which is CRF01. CRF stands for circulating recombinant forms, the hybrid virus from the original clade. So they have been introduced into China, unlike the first two, with one strain, but this time it comes with at least seven strains we call clusters. They come into different part of the country. And also they came in a very short window period between in the middle of 1990s. You can see the 7 clades in a very short window period. Why is that? And then we look at the sociological, economic factors, because China formally is closed and then it's opened, but from only one country first, Thailand. So all people, tourists, go to Thailand. At that time, it's also the peak of the Thai epidemic in the sex workers. So they brought back this virus. Why they didn't continue bring back the virus? Because Thai had a very successful 100% condom program. They knocked down the epidemic in the sex workers, so we don't introduce more virus. So that is a coincidence in the history. So this is how I describe the 3 major strains. They spread origin from across different part of the border and spread in this large country. So we can map in each province how many viral clade are there, how there is their distribution in the population, risk groups. But that is only tell you history, but it doesn't inform you about the current epidemic. So what we did for describing current epidemic to inform the intervention, prevention, so is genetic clusters. So this virus also we know as that is the genotype, but how about the phenotype? If the virus are they the same causing disease at the same speed, or are there some strains causing disease faster? We all know that they have 2 tropism of virus. Is due to 2 co-receptor binding. CCR5 or the R5 virus or CXCR4, the X4 virus. X4 virus knock down CD4 much faster. And the reason for different clade, we know AE is faster causing CD4 decline, and the CRF07 BC is not. It rarely causing the X4 virus. So we know that it's dependent by clade. How about the cluster under the clade? So that is a question we are asking. So the 3 major clade, if you look at them, the CD4 level, infected people are not that different. But if you divided them into different genetic cluster under clade, you see the difference. So for example, the clade 4, cluster 4 and 5 of AE is different. And the reason for that difference we can see is because the clade 4 has more X4 viruses. This is not only genetic prediction, but also if you isolate the virus, you test the phenotype to see the co-receptor binding and the CD4 decline in the tissue culture. They are the same, the coincidence with the X4 viruses. And the reason for this difference variation under the different cluster is because the genetic position on the V3 loop of the binding co-receptor binding region. There are 2 major binding sites, amino acid, at position 13 and 32. They played a key difference on these two clusters. And also we also identify 3 supporting amino acids. So that's the reason, because initially they are using alpha virus because the genetic variation made the cluster 4 has more export viruses binding co-receptor, and then it's knocked down the CD4 much faster. If you have $100, you should treat the cluster 4 infected people earlier, because they progress to disease faster, they die earlier. So you have to allocate resources to the faster evolving virus population. So another genetic cluster operation we make, we can link each of the person if they are transmitting virus or not genetically. So we can see at our past national survey, we can see there are 5 huge clusters, and there are several middle-sized clusters. The larger cluster, thousands of people are together. We can see 2 of them on the left-hand side, blue is the CRF07 viruses, and the 3 red ones are the AE viruses. And what is the different risk factor? The 2 07, one is homosexual, the upper one, and the low one is heterosexual and IDU, and the other 2 AE are all majorly homosexual. And also you can see where the people located in the country, you see many crossovers between the different risk factors across region. People travel very fast with the 35,000 high-speed rail. People can go to a different city in the morning and come back in the evening. So that makes the difference. And also micro level, you can also see the cluster. For example, there's a 07 BC cluster. There are 2, and they are in the southwest region. And risk factor 1, the smaller one is a homosexual, the other one is a heterosexual IDU. You can see the central located risk person, which is the senior male heterosexual guy, and this, you can see AE, the 2 clusters are homosexual, and the central located high risk person are young homosexuals, male. And also you can identify the epidemiological data by introducing the transmission cluster parameter into the data set. When you do a survey, you ask people which risk group are you belong to. What we call the self-reporting risk factor. And for example, this is in this survey we have 872 homosexual have self-reported. We have about 800 heterosexual, but divided into female and male. So the self-reporting, they are belong to the heterosexual or homosexual. But when you put the transmission factor, they didn't agree, not totally agree. The agreement is only 2/3 to their self-reporting. So what you can do is to doing another operation by using the determined model and by using machine learning process. You put the total agreement as training data set, and then you calculate through the model, machine learning, and to which true risk factor they belong to. Each time you get more people are green, you put them into the training set, you continue many times, and then you get a final true risk factor. So in this operation, you can see the homosexual, actually they are 97% homosexual. The male heterosexual, they are 100%. Self-report is right. The women are more truthful. But the problem is the male heterosexual. There are 30% of them are not real heterosexual, they are homosexual. Because the social factor, the stigma, they don't want to report. So by this way, you can really know what is the nature of your epidemic, what is your intervention, what we call precise intervention, which is needed through these research tools. And for treatment, UNAIDS WHO has a goal by this end of this year to treat like 30,000 people. They can reach their target. You see in the past history, walking towards to the red dot on the upper right corner. But the prevention, we cannot reach. If you follow the past history, we should control less than 500,000, but it will be more away over 1 million. So the prevention is a major challenge to today's AIDS epidemic. So that is why we need the precise intervention. This is also last 2 slides for China epidemic. We are still going up in the uptrend, so continuously. So this year we will identify about 160,000 new infections of this year. So that's the challenge. China also moving up with 3 major parameters. New infection, we are number 5 by all countries. Mortality, number 5. The people living with HIV is number 8. If we don't do precise intervention and to target the high-risk population, we cannot reach our goal. So thank you for your attention. Yeah.
Thank you very much for the interesting report, and we're waiting the question in written form to all the moderators of our session. Thank you. Я передаю слово.
Now I would like to give the floor to Professor Margolis, Senior Investigator, Section on Intercellular Interactions, Moscow State University Professor.
Спасибо.
Thank you. Now we move on to fundamental basic research in HIV, because it is clear that every bit of success that Mr. Moses was talking about, they all are rooted in fundamental, in basic research, as well as every fail that occurred on our way towards fighting HIV. They also occurred because we do not yet have a very clear understanding of this infection's pathogenesis. And still throughout our research, we come across absolutely unexpected discoveries. For quite a while, we've been researching HIV, we've been taking blood samples, we've been conducting experiments, thinking that we were studying a virus. Now, we see some dramatic changes in approaching HIV management and research, because now we see that uninfected cells, they also have particles that are very similar to viruses. So sometimes we don't really study a virus, but we study a mix between these virus particles and extracellular vesicles. That is a very hot topic today, because it is believed that extracellular vesicles that are produced by one cell. They have their RNA and DNA patterns, and they pass it on to other cells. And before, it was believed that only a virus cell can do that. So now there is a number of articles on this topic, and we have more and more questions. New and new vesicles are being discovered. Monthly, thousands and thousands of articles are published on this topic. Researchers discover vesicles of various sizes. So today we discovered this whole new world of vesicles, and viruses only account for a small proportion of them. And that reminds me of the discovery of the microworld. The discovery of microbes, when through microscope we saw a large number of microbes, but up to this day we do not have a clear information about some types of microbes and what kind of impact they have on the human body. As I've already mentioned, vesicles go from one cell to another, which is more sustainable than just replicating molecules, which can be compared to a bottle with a letter in the vast ocean. But vesicles are a letter sent precisely from one cell to another. But indeed, when we have such discoveries, it is very important to know who made this discovery, but we cannot be sure. What we know is the number of articles growth exponentially. In 2019, in September, there were around 80,000 articles. Yesterday, I saw the number, and there were around 19,000 of them, which shows you that it is a pretty hot topic today. Of course, we can track it all back to Charles Darwin, who believed that there are some particles of minute size generated by every cell type containing molecules, and that serve to communicate them to other cell types. We can compare it to half bubbles that are cited in Macbeth. But at this session, we need to first of all understand how they influence HIV infection. And the answer is yes, they do influence HIV. A wonderful example is the work of Mr. Sadowski, who showed that protection against HIV viruses can be passed on to various cell types through vesicles. That is closely connected to macroRNA transmission. So within a vesicle, you then have a virus protein, and T cells are activated. There is apoptosis suppression, which of course impact the infection. A couple of months ago, another article was published on bacterial EV, and you know that in our system there are more bacteria than there are cells, and they determine our immune status. So how do these bacteria work with our system? They produce extracellular vesicles, and that is how they communicate with the host system. A couple of weeks ago, we discovered that lactobacillus in a woman's vagina plays a huge role when it comes to protecting a human bodies from various pathogens and bacteria. But how this process works, it is still unclear. And now there is this fundamental research, this fundamental work that helps us to take a look into what happens in the human system. We reproduced this using specimen. And we discovered that HIV infection is suppressed, but we did not understand what helped to suppress HIV. But now we know that these are vesicles that do that. So by using these vesicles from lactobacteria, we can suppress HIV. But what do they have an impact on cells or on the virus? We believe that it will take us years to figure it out, because cells are very complex. But we were lucky because these vesicles, they did not impact cells but viruses. And viruses are a lot simpler than cells. So vesicles impacted viruses, and they inhibited viruses from interacting with cells by blocking proteins that is responsible for virus budding with target cells. Now our task is to understand the molecular mechanism behind it. It is all fine when vesicles are produced by other organisms, by bacteria. But what about a host system? That is a lot more complex, because vesicles look a lot like viruses. And it is very hard to detect them from viruses. It is extremely hard to do so, if even possible at all. Because viruses and vesicles are produced by an infected cells following the same mechanisms, by the very same particles. So how do we tell them from each other? We came up with a method that helps us to differentiate antigen content of a virus or a vesicles, because they're all different. And this overall analysis helps us to do that. But still, we need to figure out an antigen composition of each particles. And they are very small. That is why we use magnetic nanoparticles of a size of 10 to 15 nanometers. And we use that to single out various antibodies, and we are able to look into what antigens we have. And we planned to use antigens to differentiate between vesicles and viruses. But it turned out that there is no difference here between vesicles and viruses, because they inhibit the very same proteins in cells. So how do we tell a vesicle from a virus? Hopefully, out of all host cell proteins, there are some of them that are associated to HIV variants. There are some that are part of vesicles but are not part of viruses. So there are some proteins that are only found in vesicles, which helps us to tell a virus from a vesicle. So if we use a specimen that contains of a mix of vesicles and viruses, and if we use a magnet to take at least some of the vesicles out, and this is a vesicle cervix specimen, So if we take out some of the vesicles, then we will see a decrease in infection, which shows us that vesicles either activate the cells or they do something else. So our task now is to see clearly the mechanisms behind vesicles operation and how they can help us to decrease the infection in the system. So what is a virus specimen? We can look into viruses and vesicles and to look at their proteins. So what we see is that infected cells, when they produce vesicles, these vesicles have proteins like GP1 120. So if we look at markers to tell which particles are vesicles and which of them are viruses, then we see that vesicles that we see in down left corner, they have virus proteins. So what is a virus and what is a vesicle? If we take a look in a dictionary, then we will read that virus is a microscopic particle that is further developed in a cell, multiplied in a cell. But now we speak about non-active viruses and non-infectious viruses. And when we speak about HIV, 90% of this virus doesn't go into the next cycle of infections. So what makes a vesicle different from a virus? You know that biologists that do not like to give definitions, but you cannot work without giving a clear definition to a virus and to a vesicle. And we have to take a different approach. We believe there is a whole spectrum of particles produced by an infected cell. From infectious virus to exosome of uninfected cells. And there are lots of stages between these two, and we need to understand what is the correlation between them. So to my mind, this new approach to virology of HIV reminds me of physics, when throughout hundreds and hundreds of years, Newton and our physicists were speaking about visible light. And after a revolution in physics, we realized that the visible light only accounts for a very small part of a spectrum of radio waves. I believe that virology is on the edge of a very same revolution. It is especially true for HIV virology. And of course, our famous dilemma, what appeared first, a vesicle or a virus? What came first, a chicken or an egg? So, a short summary of my report. All cells, including infected cells, produce vesicles that stimulate HIV infection. Lactobacteria vesicles help to impact HIV infection, and some of vesicles have virus proteins. And I do hope that we will have the next slide on the screen. So now for us it is important to understand how vesicles and viruses are connected to have a better understanding of HIV infection, which will lead to new approaches in HIV management and prevention. And it will help practitioners such as Mr. Moses and Mr. Shao to design new tools to fight HIV and to fight HIV transmission. we are very hopeful. And the history of medicine showed us that successes in medicine are impossible without good basic research. Sir Francis Bacon said that hope is a good breakfast but a very bad dinner. But it is early in the morning, so we're all about breakfast and hope now. I work at the National Institute of Health, and of course Robert Gallo and Beatrice Vitali did so much for studying HIV infection. From Bologna University. Thank you very much. Thank you very much, Mr. Margolis, for giving us some hope for breakfast and even some hope for dinner. Now I would like to give the floor to Mr. Jürgen Rockstraw, Professor of Medicine and Head of the HIV Outpatient Clinic at the University of Bonn. Please, Professor, you have the floor.
Доброе утро. Спасибо большое for the kind invitation to speak here today. That's all my Russian. I was very impressed by Professor Margolis. Oh, right. So my task is to talk about healthcare services and take a European perspective. So what are the differences between different regions in Europe? We can go to the next slide. Yeah. So overall, it was already alluded to the high burden of HIV in Europe. You see there were 141,000 people diagnosed with HIV in 2018. Overall, 2.2 million people living with HIV in the region. And you can see that the burden of disease is in the Eastern European, Central Asian countries. And you can see that actually the number of new infections in West have come down considerably, which is a reflection of a broad implementation of prevention measures. On the next slide, you can see that the epidemic is very different in different parts of the world. So transmission factors which fuel the epidemic have been different, with the predominantly men who have sex with men infection or transmission in Western Europe, and then an increasing rate of infections due to initially intravenous drug use in Eastern Europe, which then has passed on to heterosexual transmission. Note also that in Western Europe you have a high rate of migration. Many of the countries have former colonies, so there's a lot of people coming from countries associated to these European countries, which account for the high number of infections in individuals who were born abroad, whereas this is somewhat lower, obviously, in other parts of Europe. Now, what I want to cover is really three topics. HIV testing, how does that differ? HIV care, where is that supplied? And then looking at the multidisciplinary teams, which already have been highlighted. Now, with regard to HIV testing, this can happen in public health offices, but it can also happen in community-based services, which clearly have led to changes in many parts of Europe because they are usually low barriers for entry into HIV testing. There's less stigma in these settings, but also general practitioners can do tests, and then obviously there's also now in Europe the availability of a home test. Now, if you look at the progress towards the first 90, remember that UNAIDS back in 2014 has defined 390s, which are to be achieved by the end of this year. 90% of people should know their HIV infection by the end of 2020. Of those people, 90% should be linked to care and receive antiviral therapy, and of those, 90% should be undetectable. So overall, 73% of all people with HIV should be successfully suppressed under therapy. And you can indeed see that probably the first 90 is best achieved throughout the entire European region. You can see that there are quite a number of countries which already have achieved to have 90% of their individuals being diagnosed for HIV. You can see there are some issues in some countries, but overall, I think the rate of diagnosis is relatively high. Looking at the German situation, you can see that Germany is still struggling to get the last 2% to achieve the first 90. And that tells you that even in a country where there's free access, so no one has to really pay for HIV testing, stigma remains the biggest barrier because people are afraid of going to testing, afraid of disclosing their sexual preferences or committing that they're using drugs. And that is really, I think, remains a barrier, even in a country where health insurance-wise pretty much everything is possible. And even though we do have general checkpoint implementation, so we have community-based testings, but that does not guarantee that everyone gets tested. You can see that of those who are tested, there is great linkage to care, which has a lot to do with the general availability of health insurance coverage. And you can see that the people who are receiving antiviral, 95% are suppressed, really highlighting how successful modern antiviral therapy is these days. Whoops. This is a sort of a joker slide. Oops. Back. We just, no, you have to go back like, yeah, one more back, one more back, and one more back, and one more back. Thank you. So what is the big change now in the German setting? And I think this is the only way we are going to able to get our first 90 up. And the one is the introduction of the home test. So you can buy an HIV test now in a drugstore, in a pharmacy, and get tested. Something which already has been available in the US for quite some time, has now also made its way into the German scenario. And I think it's important because it allows complete anonymous testing for people who are very stigmatized and do not want to disclose to their family. I think this isn't an additional option. And then obviously the registration of pre-exposure prophylaxis, so the prophylactic intake of antiretroviral drugs which work against HIV in people who are HIV uninfected to prevent transmission, is the big changer because it now allows people to take identity of their own sexual health and to get regular testing. And clearly pre-exposure prophylaxis is the big changer in those countries where it has become available. And I will show you data which was presented at the recent European AIDS conference from France, which in major cities like Paris has signed a fast-track initiative strategy which implies to lead to an increase in testing increased in testing facilities, but also trying to establish testing programs we agree. Yeah. So they've also established testing programs for difficult to treat people. So even in any given society, you will have groups which are particularly vulnerable, including migrant MSM, black MSM, and other social and ethnic minorities. And to specifically offer testing for these groups, I think will help to close the gaps. And what this extended testing has led to is actually a decline in new infections, which means that more people have been diagnosed, more people are put on therapy and successfully suppressed. And in particular, the introduction of PrEP has led to a 28% reduction in new infections among MSM born in France who take these medications. So again, showing you that the introduction of PrEP actually leads to a sharp decline in new infections. But also note that MSM who are born outside of France, so in particular North African ones, see no change, which is due to less access in the healthcare system in France. So clearly that is where they need to improve their efforts. Now, the wonderful thing about HIV 2020 is that life expectancy has really become almost that of the normal general population. And this is just one example from the Swiss cohort because they're particularly well organized and capture all their HIV individuals in a large national cohort and match the life expectancy to the general population. And you can see that with the introduction of better tolerated drugs, longer half-lives, single tablet regimens, the life expectancy has become very close to the one in blue, and we all know that in Switzerland, life expectancy is particularly good because you have free access to chocolate and mountains and great air. So obviously, treatability has become great. The question is, how can we get everyone on treatment? And probably most importantly for the patients, something has changed in the simple message that if you become undetectable under therapy, you cannot pass on the virus. So undetectable equals untransmittable, and it's really, I think, the greatest motivation for patients to stay on therapy. And if you look at the somewhat dismal response rates in vulnerable groups, particularly IV drug users, at least in my setting, I've learned that if I spread that message very strongly, I can get people motivated and convinced to stay on their therapy. So don't underestimate the impact this has, because no one wants to infect their partner. Now, there's something to be said about the training of physicians. So there have been various papers, and this is just one example, looking at the relation of physician specialty and experience in treating HIV patients. And one of the ways to look at that was to say who chooses guideline recommended antiviral therapy, and obviously that worked better in ID specialists, so people who were specifically trained for practicing infectious diseases, but worked less well in internal medicine specialists who had low experience with HIV. So HIV care does require a certain amount of expertise, and I think that's important to highlight. Now, as the current president of the European AIDS Clinical Society, I'm very proud to say that we have just sort of launched our latest version of the EX guidelines, and they're really trying to help all countries in Europe to sort of go towards the same standard of care. And you will find in this book, which has really increased rapidly and is very big by now, a very detailed way of how to assess people living with HIV and to really look at all the different aspects. And clearly HIV does not only come as a medical condition, but has a lot of psychosocial issues, sexual reproductive health issues attached to it, which also need to be addressed. And you will find in this booklet all the various things you need to look at, including all kind of co-infections, other sexually transmitted diseases. Don't underestimate the role of viral hepatitis co-infection. Obviously, tuberculosis also in this region, very important. But then also in an aging patient population, many different comorbidities, including renal disease, hypertension, diabetes, and so forth. I think what is reassuring is to see that European countries are sort of getting much closer. The ART, so when do we start therapy, policy has become very close. Almost all guidelines now do recommend immediate initiation of antiviral therapy once diagnosis is established, on the basis of the wonderful START study, which showed a benefit even with high CD4 count. So you can see how over the last years this has transitioned to universal recommendation for immediate ART initiation. And as a function of that, I think we see much more people being on HIV therapy. So I think we have to also look at this positively, that we are increasing the amount of people on ART, even if that could be further improved substantially. Looking at the third 90, I think it becomes clear that we are far away, and Professor Mazuz has highlighted that it will be probably very difficult to achieve. But I think this is the point where we all need to work together and see what has worked successfully in certain places and can be implemented in other places as well. And that really brings us to the multidisciplinary teams, and both of our Alexeys, Professor Aramov and Professor Mazuz, have highlighted the availability and the interlink between services in the Moscow region. I think this cannot be highlighted enough, that in particular in an epidemic where IV drug use is a main driver, you will need close links to social workers, to psychologists. The rate of psychiatric comorbidity is very high. I think that drug dependency services are also important to emphasize. Clearly in some of the southern European countries like Spain and Portugal, the offering of substitution with methadone as well as clean needle exchange programs have substantially eliminated new infections among IV drug users, to highlight that particular point. But in patient populations which is struck by comorbidities and co-infections, it is very good to work together with the hepatologist and with other related disciplines, and really, I think multidisciplinary teams are key in the success of maintaining viral suppression and offering best care for these individuals. The number of patients who are aging is obviously high, and you can see here from our own hospital database that the amount of patients who are above 50 is almost now approximately half of all our patients. And so in this aging patient population, different things need to be considered, and you see here in a sort of a diagram all the different issues which need to be considered in an older HIV-infected patients. Besides the social factors, which always play a role, there obviously are HIV-related factors, in particular chronic inflammation, even the setting of viral suppression, which probably accelerates some of the comorbidities, in particular cardiovascular disease, but also think of the increased rate of bone fractures, renal disease, COPD, and the most common cause of dying now in an HIV-infected individual being non-AIDS defining malignancies. We also need a stronger interaction with oncologists and screening programs for cancer entities correspondingly. And then obviously there are a lot of issues around the antiviral therapy we give, and don't forget the risk of drug-drug interactions. So in an attempt to sort of help to implement better care throughout Europe, we are working on standard of care meetings where we invite people from all the different European regions, but also institutional partners such as ECDC, WHO, UNAIDS, and HIV in Europe to really kind of try to implement better standards of care. The last meeting was around 3 key areas: diagnosis and treatment of hepatitis C, HIV testing, late presentation, and TB. And we've started to launch audit projects where we sort of ask general questions. Do you have hepatitis vaccination? And those who test negative upon hepatitis serology, for example. And this first pilot was conducted around hepatitis issues. And indeed, what you can see in the outcome, for example, when we asked the question of was hepatitis delta checked in people who have chronic hepatitis B. Remember, hepatitis delta is a superinfection and needs to be checked for. You can see that this happens in some centers in everyone, but in some centers it doesn't happen at all. So I think that is where there's a need to sort of tie things together and implement better strategies based on the guidelines, and to do audits and then repeat the audits and see how things change over time. So I think there's a huge effort ongoing to streamline and improve the standard of care in Europe. So in summary, I think HIV testing strategies need to include all possible interventions, including really community-based checkpoints and community-based offers, because that at least allows a lower barrier into the testing environment. I think we need to improve training of physicians and increase access to HIV care. I think we have to be well aware that people living with HIV are living longer, so we have to also focus on healthier living and deal with multidisciplinary approaches to cover all the comorbidities which may occur. Develop treatments and interventions specific to people living with HIV. We have to develop a European standard of care, perform audits, and improve overall all our efforts together. So with that, I'll close and just say every action counts. That's the slogan of the European AIDS Society, and thank you for your attention.
Thank you very much. It was really very interesting to receive the information about European approach and the possibilities to end the epidemic of epidemic. Now we have only 10 minutes for the plenary session, and I think we could pose all the questions to our guests Emin Shao and to Alexey Mazus. He's not the guest. At future sessions, which we would have during this assembly, but our guest Leonid Margolis can answer all the questions at this session. И я передаю слово Елене Валерьевне Цыгановой для завершения.
And now let me give the floor to Ms. Tsyganova. If there are any questions, first of all, let's ask Professor Margolis first.
Dear colleagues, first of all, let me please briefly speak about our international educational center established with the Russian University and Moscow AIDS Center. So what do we offer to our students? We offer simulation education because the Russian University has a very state-of-the-art stimulation center. We also offer programs in healthcare management and clinical studies of HIV infection. Starting from 2013, we held weekly seminars, workshops. Several times per year, we held conferences and webinars with international experts. That is why we have expertise in this matter. We also offer studies of co-infection and studies of other infectious diseases. And now the questions to our expert. Mr. Margolis, how do you think we can defeat HIV infection from the point of view of fundamental basic studies? You know, that is a very nice question. Well, first of all, I'd like to tell you that if you have my email and we do not have time to answer all of your questions now, please write to me and I'll answer you. So how can we defeat HIV? A nice question, and I think it can also be answered by other experts at the table. Well, first of all, I believe partly we've accomplished the task because several years ago it was hard to imagine that we would have 30 medications to choose from to suppress HIV. And we know now that people living with HIV, if they receive their ART therapy, they live as long as HIV-negative people. So to some extent, we have defeated HIV. But how do we defeat this whole epidemic? To do that, we need to have a better understanding of how HIV is transmitted. We think we understand that, but we are actually really far from this full understanding. And if we have vaccines that will help us to put an end to transmission, then we will defeat HIV. Because today, despite the fact that HIV-positive people live longer, they tend to age quickly, they have cardiovascular diseases, they sometimes suffer from diabetes. So there is a lot of room for improvement. So that is why basic research and clinical research should focus on tackling transmission from one person to another. Thank you so much. There is one more question to you. So, Mr. Margolis, what are vesicles? Are they organisms in themselves or they're byproducts of a virus? Neither a vesicle nor a virus are organisms per se. I believe we can go in depth discussing what these are, but they are particles. They are no organisms per se, because they need cells to multiply. And cells, they are living organisms. The next question goes to Professor Shao. In your opinion, what mechanism is responsible for more rapid progress of HIV in case of CXCR4 tropism?
In our study, we found that the genetic makeup of this cluster, we still don't know why is that. They have the threshold for what we call co-receptor shift is being lowered. So they are very fast making the co-receptor shift from R5 virus to X4 viruses. We already identified the two major positions on the V3 loop, and the 3 supporting positions, amino acid on the V3 loop. But why they evolved into that direction, we still do not know. But think about other viruses like causing diarrhea, echo, or they have about 100 subtypes. But HIV subtype, we have only 2, HIV1, HIV2. Since HIV has been in human in less than 100 years. So for other viruses, there are 1,000 years.
But the question was why you think shift of is associated with the acceleration of progression?
Because the X4 virus, they cause syncytial formation. Syncytial formation, they have been killing the cells without virus. So they accelerate the cell killing. That is the main mechanism.
Вопрос, наверное, несколько философский.
Now we have a somewhat philosophical question to our expert. Do you believe there is an opportunity to create a prevention vaccine? And if so, who will have to take this vaccine?
Who wants to take the responsibility?
Okay, since I'm working on vaccine almost 30 years, maybe I think I can shed some light on that. I think HIV vaccine is very difficult because of we have vaccines against the disease who are immune system in evolution are stronger than the pathogen. But HIV, since it's a very new virus, our immune system have not adopted. In natural infection, we do not have a protective immunity developed, unlike other viruses like HIV, hepatitis. For people who should take, I think the people who have a risk, it has been evolving. Early epidemic 30 years ago, many MSM and hemophilia patients. But now, then we have IDU patients are the risk group. But now, in certain countries, like in your slides, in Eastern Europe, heterosexual is a major proportion of HIV infection. So for heterosexual, almost all humans have heterosexual activities. So depending on the risk level, we don't need to vaccinate all people. But we start with the high risk first, and then we can move to the middle risk. If that is already can contain the virus, we don't need to further vaccinate the people because HIV is easy to prevent. And also health education before vaccine. Health education, changing behavior is the best vaccine.
We know whom to vaccinate, we don't know what we vaccinate with.
And also there's some side effect of vaccination. If you have a vaccine for HIV, you prevent HIV transmission sexually. But other diseases cannot be prevented, like maybe syphilis, gonorrhea can increase if you have an HIV vaccine.
But let me also highlight that probably the vaccine, if it should come, will not have 100% vaccine protection. So we should also know that we're competing with other prevention interventions like PrEP, where if you take PrEP regularly, you almost have 100% prevention. So that, I think, is one of the big challenges in vaccine development today, is that there are now prevention interventions which are almost 100% successful. And the only trial which showed a little bit of effect was the Thai vaccine with 33% protection. So that's obviously not enough. And I agree that once you have the feeling you're protected, but you have a remaining risk, this is going to be a complication.
Уважаемые коллеги, к сожалению, время нашей сессии заканчивается.
Dear colleagues, unfortunately, we're running out of time. You still can pass your questions on to me, and I believe that during our next sessions we'll be able to answer them and to listen to some very enlightening answers.